A simple pregnancy test, which can be done at home today, used to need a laboratory visit a couple of decades ago. Advanced technologies have made it easy to measure and monitor health parameters such as blood sugar, blood pressure, and oxygen concentration at home. While simplifying testing and monitoring on one side, biotechnology and medical engineering also bring advanced and effective methods such as CT and MRI scans, genetic engineering, and biocompatible implants to diagnose and treat severe conditions and diseases. Innovations from the Indian Institute of Technology Bombay are adding their share to make healthcare better, affordable, and accessible. Professor Rohit Srivastava's Nanobios laboratory works on developing portable, cost-effective, and easy-to-use diagnostic devices for testing glucose, thyroid, cholesterol, and other health parameters. They have developed a compact and easy-to-use lipid analyzer.
The lipid analyzer consists of a reader and a strip. The strip has 3 testing pads. Using 1 drop of blood, I can tell you total cholesterol, HDL cholesterol, triglycerides, and LDL using a formula. This is a unique product because this product can tell you your lipid profile in less than a minute at point of care.
Professor Srivastava's lab also developed a device to check thyroid markers T3, T4, and TSH in the blood.
Now this device has a reader as well as a strip. This device uses a standard lateral flow immunoassay strip.
A lateral flow strip is used in a home pregnancy test kit. The test is positive if the test line turns red.
Now using the same principle, if I can have my analyte being tested on the test line, the intensity of the test line will tell us how much of the analyte is present in the sample. These devices are going to be extremely useful at point of care because when placed in tier 2, tier 3 cities and small pathology labs across the country, one, they will significantly reduce the cost of testing. Second, they will actually make results available for the clinician to make a diagnosis as soon as possible.
Professor Debjani Paul's Microfluidics and Biological Physics Laboratory uses microfluidics to build point-of-care diagnostic solutions. The team has developed a technology to measure the stiffness of a single cell. Stiffness of cells may change in certain conditions such as infection, oxygen deprivation, or cancer.
Conventionally, the stiffness of single cells is measured using optical tweezers, micropipette aspiration, or atomic force microscopy, and so on.
These methods are expensive, have low throughput, and need an elaborate setup.
This is where microfluidics comes in, and we can measure stiffness of tens of thousands of cells over a long period of time. What happens in this device is a cell comes along a straight channel and then it faces a pillar, and after facing the pillar it either goes left or it goes right, and how far it is from the pillar after it passes the pillar decides whether the cell is stiffer or whether the cell is softer.
The same technique can also be used to check if stored blood is suitable for transfusion.
Sometimes the blood has no infection, the hemolysis is less, and still the blood cells become stiff, in which case they should not be even transfused. Before every transfusion, they can measure the stiffness of the red blood cells and they can see if the blood can be given. Going forward, we would like to move on to other kinds of cells, for example, cancer cells. There are certain cancers where the cells become stiffer, and our device could be a very interesting way to look at the stiffness of many cancer cells in a high throughput manner.
Professor Hari Verma's theoretical and experimental bioimaging laboratory uses light as a probe for non-invasive brain imaging in animals and humans. The technique is called near-infrared spectroscopy or NIRS.
The idea of near-infrared spectroscopy is to illuminate the tissue with certain light in the wavelength range of 600 to 1000 nanometer.
Near-infrared spectroscopy is used in the pulse oximeter, a small device that fits on your finger and measures the oxygen saturation in your blood. It uses an LED to illuminate the tissue. The light scattered by the tissue is measured to derive oxygen saturation.
We use the same principle to look at the human brain to measure the cerebral metabolism in the human brain. The light is delivered to the brain via the forehead using optical fibers. The scattered light from the deep tissue is measured using different optical fibers connected to very sensitive optical detectors. From this data, we can do a data fitting using light diffusion equations to measure the cerebral metabolism. So there is a variant of near-infrared spectroscopy called diffuse correlation spectroscopy. I've created as DCS.
DCS uses a laser instead of an LED. Using DCS, one can measure blood flow in the brain non-invasively. When combined with NIRS, we can observe the blood flow and metabolism in the brain together, facilitating appropriate diagnosis and treatment in brain-related disorders.
The advantage is the simplicity in instrumentation. So if you look at the CT scan or MRI, a bedside monitoring of the patient is not possible. But with this optical technology, we can actually do a bedside monitoring. Second thing, it gives you information about molecular signatures. Say for example, you can look at the oxygen concentration, right, by looking at the oxy and deoxyhemoglobin concentrations, which is not possible with a CT or MRI.
Professor Prakriti Tayalia's Cell and Tissue Culture Laboratory develops implants for cancer gene therapy. Gene therapy for cancer uses lentiviruses to modify immune cells that can then kill cancer cells. Lentiviruses deliver DNA that create the desired gene modifications used for therapy. In conventional gene therapy, immune cells are derived from the patient, modified in the laboratory, and injected back into the patient.
Implant-based cancer immunotherapy is another approach of cancer immunotherapy where we are using an implant or a scaffold to direct the immune cells towards a particular action or direction. The implant is modified with those lentiviral vectors, implanted into the body, and then the immune cells reach out to those scaffolds, they get modified, and then they move towards the tumors to do the anti-tumor action.
The implants are macroporous, providing a large surface area for the lentiviral vectors and easy access to host immune cells. The implant needs to be surgically placed next to the tumor. In case the tumor is not accessible, the implant can be placed next to the nearest lymph node. Professor Tayalia's lab also works on tissue engineering.
Tissue engineering is an approach where we use the materials, cells, scaffolds to engineer an organ either inside the body or outside of the body for wound healing or replacement of a particular organ.
The team has developed a bilayer bandage for healing wounds. The inner layer is macroporous and absorbent to absorb the wound discharge. The outer layer repels water and keeps infection-causing microbes away. This bandage can be effective in healing burn wounds. As researchers at IIT Bombay continue developing novel biosensors, drug delivery systems, imaging platforms and materials, they promise to bring new advancements to healthcare in India.